Good morning to all my dear students. Welcome to our Zoology online class. The last class, molecular genetic lesson, we have studied about an important topic, very important 5 mark question, translation. Okay, so once again repeat the definition for this word translation. See, translation meaning is the process of polymerization of amino acid to form a polypeptide chain is called translation. Okay, you all know the polypeptide chain is playing a very important role in the formation of protein molecule. So the process of polymerization of amino acid molecule to form a polypeptide chain is called translation. And this translation process takes place in 3 steps: initiation, elongation, and termination. Okay, so last class we studied about this translation. Okay, friends, today we are going to study about 2 more important topics. Okay, the first topic is called opera. The first topic is called opera. Here, opera means a cluster of genes. Okay, a cluster of genes doing a related function. Okay, a cluster means a group. A group of genes performing or doing a related function is called opera. Okay, and here this operon is a unit of gene regulation and gene expression. Okay, operon is a gene regulation unit, is a unit of gene regulation and gene expression. And this operon molecule consists of 3 important parts. One is a structural gene, second is a promoter, and third one is operator. So a operon consists of, operons means a cluster of genes performing a similar function or related function is called operon, and it is a unit of gene expression and gene regulation. Okay, then it consists of 3 important parts. Okay, the first one is the structural gene, second one is a promoter region, and the third one is the operator region. Okay, so here the structural gene, one or more than one, okay, one or more structural genes are present in a opera. Okay, and these structural genes are playing a very important role for the production of protein, tRNA and RNA. Okay, so in the operon, the structural genes function is coding for protein molecule, coding for the rRNA, coding for the tRNA required for the cell. Okay, and one or more structural genes are present in a operon. And the second region is called promoter region. This promoter region is a sequence in a DNA molecule. Okay, and this sequence only initiating the RNA synthesis. Okay, this sequence only initiating the RNA synthesis. So promoter region is one of the sequence or some of the region of a DNA molecule, and this region only initiating the RNA synthesis. Then next, the last portion, the last part is called, the region is called operator region. This operator region is present between the promoter and structural gene. Okay, between the promoter and structural gene. Okay, that's all about this operon. It's one of the very important 3-mark questions. So once again repeat, operon is a, it's a structural, it's a unit or unit of gene regulation or and gene expression. Okay, a group of genes, okay, or a cluster of genes doing a related or similar function is called operon. It has 3 important regions. The first region is called structural region, structural gene, and the second one is called promoter, and the third region is called operator region. Okay, my dear students. Then now we are going to study about one of the very important 5-mark question: structure of lag operon. A structure of lag operon. Listen very carefully. Very important 5-mark question. Say, in Escherichia coli bacteria, lactose metabolism is one of the important process. Okay, and for this process, lactose metabolism process, Escherichia coli bacteria need 3 important enzymes. So there are 3 important enzymes controlling the lactose metabolism in Escherichia coli bacterial cells. Okay, the first enzyme name is called permease. Okay, first enzyme name is called permease. The first enzyme name is called permease, and the second enzyme is called beta characterization. Okay, beta galactosidase. And the third enzyme, so first enzyme is called permease, second enzyme is called beta galactosidase, and the third one is called transacetylase. Okay, so these three enzymes playing a very important role in lactose metabolism in the Escherichia coli bacteria. Okay, so here this permease enzyme is very important. This permease enzyme is needed for the entry of lactose inside the bacteria cell. So the function of this permease enzyme is this permease enzyme only allowing the lactose molecule inside the Escherichia coli bacteria cell. Then second, the function of this beta galactosidase. This beta galactose enzyme is used to convert the lactose into glucose and galactose. Okay, so the function of beta galactosidase enzyme is converting or dividing the lactose into glucose and galactose. Okay, then the third enzyme is called transacetylase. This transacetylase enzyme converts the acetyl-CoA into beta galactosidase. Okay, so now I said three important enzymes names and the functions of these three enzymes. So the first enzyme name is called permease. The function is this enzyme is needed for the entry of lactose inside the bacterial cell. And the second enzyme beta galactosidase. This enzyme convert or hydrolyze or break down the lactose into galactose and glucose. And the third enzyme transacetylase. Okay, this enzyme convert the acetyl-CoA into beta galactosidase. Okay, my dear students, I hope you understood all this point. Then the next point about this lactopron is, see, the synthesis or the production of these three enzymes are controlled by three important genes. Okay, three important genes. For example, say this permease, the synthesis or the production of this permease is controlled by a gene. Name of that gene is called a LAC-Y gene. Okay, LAC-Y gene. Okay, then the production of this beta galactosidase is controlled by a gene is called a LAC-Z gene. Okay, then the production of this transacetylase is controlled or regulated by a gene called LAC-A gene. Okay, so Y gene, Z gene, A gene. So this LAC-Y gene is coding for this permease enzyme, LAC-Z gene coding this beta galactosidase enzyme, and LAC-A gene coding this transacetylase enzyme. Hope you understood all these points. So this point is very important for 2 mark questions, because they will ask one question: what are the enzymes needed or playing a very important role in the lactose metabolism in Escherichia coli bacteria, and what are the functions of that enzymes? Like that they will ask a question. Okay, then say this lactopron consists of 3 important regions. One is a regulator region, otherwise called I region or IG. Okay, a regulator gene, otherwise called IG. And the second region is called promoter site, otherwise called P site. And the third region is called operator site, otherwise called O site. Okay, so the second point about this lactopron is, this lactopron consists of a regulator gene. Name of that regulator gene is called IG. Then it has 2 sites. One site is called promoter site, otherwise called P site, and the second site is called operator site, otherwise called O site. Understand, my students? So I hope you all understood these points. Then 2 scientists, Jacob and Monod. Okay, these 2 scientists only first developed a model for this lactopron. Okay, so Jacob and Monod, these 2 scientists only first developed a model for this lactopron. Okay, so here in Escherichia coli bacteria, When the bacteria gets or receive its normal energy source glucose. Listen very carefully. So when a Escherichia coli bacteria receive a normal energy source glucose, this I gene, okay, this I gene transcribe a mRNA molecule. I gene transcribe a mRNA molecule. Then at the end of this translation process is over, a protein is produced. Name of that protein is called repressor protein. Name of that protein is called repressor protein. This repressor protein bind with the operator region. Immediately the translation process is prevented. So once again repeat this point. In Escherichia coli bacteria, when the Escherichia receiving or getting its normal energy source glucose molecule, this I gene transcribe a mRNA molecule, and at the end of the translation process, the mRNA molecule producing a protein called repressor protein. This repressor protein bind with the operator region. Immediately the translation process is prevented or the translation process is stopped. So this is happening when the bacterial cell receive the normal energy source. What is the normal energy source here? Glucose. Okay. So in the absence of glucose, when the glucose molecule is absent, the Escherichia coli bacteria using lactose instead of glucose. Okay, so in the absence of glucose, the Escherichia coli bacteria receiving or using the lactose as their energy source. Okay, so this lactose acts as a inducer. This lactose acts as a inducer. So this lactose interact with this. Okay, so this lactose interact with this repressor protein. Finally, the repressor protein is inactivated. So once I repeat this last point. See, if the glucose molecule is absent, Escherichia coli using lactose for their energy requirement or energy needed. Okay, so when this lactose molecule is entered inside the bacterial cell, already I told you this permease enzyme only allowing this lactose molecule inside the bacterial cell. So with the help of this permease enzyme only, the lactose molecule is entering inside the cell, and the bacteria now using this lactose for the energy purpose. Okay, so when the lactose is entered inside the cell, it acts as a inducer. That means it interacts with the repressor protein. Immediately the repressor protein is inactivated. The repressor protein is inactivated. Understand a few? Now, see, now this repressor protein. Okay, so the repressor protein is finally inactivated. Now, RNA polymerase is produced. This RNA polymerase produces more amount of a special type of mRNA is called lac mRNA. Special type of mRNA is called lac mRNA. So this lac mRNA produces all the enzymes needed for the metabolism of lactose. Listen very carefully. So in the second point I said, when this lactose molecule is entered inside the body of the bacterial cell, immediately it is interactive with the repressor protein. Now the activity of the repressor protein is stopped or the repressor protein is inactivated. Now RNA polymerase enzyme, okay, now RNA polymerase enzyme bind with the promoter region and produce more amount of a special type of mRNA is called lac mRNA. This lac mRNA only producing all the other enzymes needed for the lactose metabolism in a bacterial cells. Understand a few? Understand? So, this metabolism, this regulation of lactopron by a repressor is an example for negative control of transcription. It's an example for negative control of transcription. But at the same time, this lactopron also under the control of positive regulation. Okay, so this regulation of this lactopron by a repressor is an example for negative control of transcription. But the lactopron also under the control of positive regulation. Understand a few? That's all about this lactopron. So lactopron is one of the important transcription unit. Okay, it's a unit of gene transcription and gene replication expression. Okay, so what are the enzymes present in this lycopron? There are three enzymes: permease, beta carotenoids, and third one is transcriptase. This enzyme's function is this enzyme only allowing, or this enzyme is needed for the entry of lactose inside the bacteria. And beta carotenoids, this enzyme hydrolyzes the lactose into glucose and galactose. And this trans-acetylase enzyme converts acetyl OA into beta galactosidase. Okay, so these three are the enzymes. And three regions present in a lycopron. The first gene is called regulator gene, other is called IG. And the second one site is there called promoter site, and the third site is called operator site. Understand of you? So I hope you all understood this 5 mark question. Okay, it is one of the very, very important 5 mark question. Then our next topic is our next topic is DNA fingerprinting. DNA fingerprinting. See, it is one of the very important technique playing a very important role in many fields. The biological field, medical field, environmental protection field. Okay, so like that, this DNA fingerprinting technique playing a very important role in many fields. Okay, so now we are going to study about this DNA fingerprinting technique. This DNA fingerprinting technique was first developed by one scientist. His name is Alex Jeffrey. Okay, Alex Jeffrey. Then he developed this technique in the year of 1985. Okay, in the year of 1985. See, normally the chemical nature of the DNA, okay, in all the human beings is same. In each and every human body, the chemical structure or the chemical nature of the DNA molecule is similar. But there are millions of differences are present in the DNA sequence of a base pair. So chemically the DNA molecule, the chemical nature of the DNA molecule is same in everybody's body. But the chemicals, there are lot of millions of differences are there in the base sequence. Okay, so because of this reason, all the human beings are different from each other except identical twins. So let's repeat this point. The chemical nature of the DNA molecule is similar in all the human beings, but there are millions of differences are there in the base pair sequence. Okay, so because of this reason, the human beings, everyone are different from each other except the identical twins. Okay, then you all know there are 23 pairs of chromosomes are present in our body. 46 chromosomes are present, and all these 46 chromosomes are arranged in 23 pairs. So inside our body cell there are 23 pairs of chromosomes are there. Then totally more than 1.5 millions of genes are there. Okay, totally more than 1.5 millions of genes present in 23 pairs of chromosomes. Okay, understood? Then inside our body cell, some segments of the DNA molecule is code for protein. That means they are playing a very important role in the protein synthesis. So, okay, some segments, not all the segments, some segments of a DNA molecule code for the protein. Then some segments are doing a regulatory functions. So some part or some segments of a DNA molecule doing the regulatory functions. And some sections or some segments are act as a intervening sequence, and some segments are act as a repeated sequences. Okay, so a large DNA molecule is there. It is divided into many segments. So some segments coding or responsible for the production of protein synthesis. Some segments are doing regulatory functions, and some sections are called intervening segments, and some segments are called repeated segments. Okay, then in our DNA molecule, a specific sequence or a specific region is there. Okay, so this specific region is called repetitive nucleotide sequence. Repetitive nucleotide sequence. This repetitive nucleotide sequence is also called VNTR sequence. VNTR sequence. Okay, see, variable number tendon repeats. Okay, variable, listen very carefully, the full form, the variable number tendon repeats. Okay, once I repeat this last point, in our body cell, a specific sequence is present in the DNA molecule. It is called repetitive nucleotide sequence, otherwise called VNTR sequence. Okay, VNTR sequence. Same. DNA fingerprinting involves identifying the differences in some region of the DNA molecule. Differences in some specific region of the DNA molecule. And this specific region is called repetitive DNA. This specific region is called repetitive DNA. Also, I repeat this point. In DNA fingerprinting technique, okay, this DNA fingerprinting technique involves the identifying the differences in some specific region of a DNA molecule, and this region is called repetitive DNA. Okay, and in this repetitive DNA, a short portion of the DNA molecule is repeated for many times. A short portion of the DNA molecule is repeated for many times. So that really this region is called repetitive DNA. Okay. Then this repetitive DNA is separated from the bulk DNA. Here bulk DNA means other region, except this repetitive region is called bulk region. Bulk DNA. Okay. So this repetitive DNA is separated from the bulk DNA. Okay, so the small portion, so bulk DNA is there, then the small portion DNA, small part of the DNA molecule is called satellite DNA. Okay, so in a DNA molecule, many segments are there, already we have studied. So some segments have some differences. So these segments are called repetitive DNA. Okay, it is present in one DNA. Then the other region is called satellite DNA. Other region is called satellite DNA. See, then depending on the number of base pair present in a DNA molecule, then depending on the length of the DNA molecule, okay, depending on the number of base pair, then depending on the length of the DNA molecule, then depending on the length of the satellite DNA. Okay, sorry, depending on the length of the DNA segment. This satellite DNA is again classified into two types: microsatellite DNA and mini satellite DNA. Listen this last point very carefully. See, based on the number of base pair, based on the repetitive unit, then based on the length of the DNA molecule, the satellite DNA is divided into two types: mini satellite DNA and microsatellite DNA. These two types of DNA molecule, DNA segments, they will not encode for any protein molecule, but they are playing a major role in the human genome. Okay, so this microsatellite DNA segment and the mini satellite DNA segments, both segments are not playing any role in the protein synthesis, are not coding for proteins, but they are playing a very important role or very important portion in a human genome. Then this DNA molecule, for this DNA fingerprinting techniques, maximum the DNA molecules are taken from the blood, hair or from the skin cells. Okay, so these DNA molecules are used for many purposes. For example, used to identify the criminals. Okay, used to identify the criminals. So for DNA fingerprinting technique, the DNA molecules are taken from the blood or hair or the skin cells, and it is compared with this VNTR segments. Okay, compared with the VNTR segments. So by comparing this with this VNTR segments, the doctors or the scientists will identify the criminals. Okay, understand? Okay, my dear students. Then finally, we are going to study about the uses or the applications of this DNA fingerprinting technique. See, the first use or the first application of this DNA fingerprinting technique is this DNA fingerprinting technique playing a very important role in the forensic science analysis, okay, or forensic science. Say, so with the help of this DNA fingerprinting technique, we can identify the people, those who are involved in criminal activities. So the criminals are identified by using this DNA fingerprinting techniques, particularly paternity and maternity dispute, okay, father and mother problems. Okay, so these disputes can be analyzed or can be cleared with the help of this DNA fingerprinting technique. So the first application of this DNA fingerprinting is what? This forensic analysis. Then second one is pedigree analysis. So with the help of this DNA fingerprinting technique, the genetic disease in the human beings can be detected. So in our the human beings are affected with different types of genetic diseases. Okay, so the genetic diseases are identified with the help of the pedigree analysis. So this DNA fingerprinting techniques playing a very important role in pedigree analysis. Second use. Then the third use is conservation of wildlife. That means this DNA fingerprinting technique is used to protect the endangered species. Okay, so the DNA information, okay, about an endangered species is protected. If a DNA sequence of an endangered species is protected, we can protect that endangered species population with the help of this DNA fingerprinting technique. Okay, so the endangered species is protected with the help of this DNA fingerprinting technique. And the last one is called anthropological studies. That means with the help of this DNA fingerprinting technique, we can analyze the origin and the migration of the human population and the genetic variations of the human population. Okay, so now I said 4 important applications of this DNA fingerprinting technique. The first one is forensic analysis. So in this field, in forensic analysis field, we can identify the criminals by using the DNA fingerprinting technique, particularly paternity and maternity dispute. Then second use is it is used in the pedigree analysis. That means it is used, the DNA fingerprinting technique is used to analyze the genetic diseases. It is used to detect the genetic diseases. Third one is conservation of wildlife, meaning it is used to protect the endangered species. Then the last one is it is used in the anthropological studies, meaning the DNA fingerprinting used to understand or used to study the origin and migration of human population and the genetic variations in human population. Okay, my dear students. So today we have completed 2 important 5-mark question and 1 important 3-mark question. Okay, 3-mark question is structure of folklore. The structure of folklore is small question. Then 2 5-mark question. The first 5-mark question is word question we studied. See, this DNA fingerprinting. Okay, DNA fingerprinting is very, very important 5-mark question, and the next question is structure of folklore. Okay, okay, my dear students. So I hope you all understood today's class. Study well and write the answer today itself and send them to my WhatsApp. Okay, okay, we will see in the next class. Thank you.